Last week, or you can pass with your neighbor. Then I will list some of your learning from last week. Okay, there's something about hemostasis. Okay, 2 points. So recap with your friends, what are the components of blood? What do you find inside blood? Number 2, what are the mechanisms of hemostasis? How do we stop our cells from bleeding out? Okay, ready? This morning, what are the components of blood we learned last week? Those are toppers, you can give me the straightforward one also. Yeah? Okay, Henry, give me one component of blood.
Red blood cell.
Yeah, okay, give me the easiest, right? Red blood cell. Okay? You right? White blood cell. Okay, and then we started venturing into other components that we may not have heard before as a student. Wednesday? Plasma. Okay, thank you. Plasma is a very liquid that all of our cells, blood cells, are floating in. Really yellowish in color. Lots of dissolved substances also. Anything else? City. Platelets, thank you. And we find that platelets play a very important role in hemostasis later on. What else can we find inside our blood? Sorry? Proteins, okay? You can find lots of proteins inside our blood. Some of them we've learned before, some of them we will learn today. Okay? If someone were to ask you which component of blood is very essential for the transportation of oxygen, which one would it be? Red blood cells. What is inside red blood cells that allows our red blood cells to carry oxygen? Hemoglobin. Okay, so that's one part settled. How do we transport carbon dioxide? Maybe let's answer the first one. Where do we transport carbon dioxide lastly? Out of all of these fellas. Plasma. So this is where we mainly transport CO2. Okay, so here is where we mainly transport O2. Here is where we mainly transport CO2. But not in the form of gas, because you know these gases are very insoluble in water. So how? How is our CO2 able to dissolve in plasma? Okay, thank you. What you want to know, right? Carbonic anhydrase. You can find this enzyme somewhere. When you find this enzyme, you find it in the red blood cell. Okay, it comes to convert your CO2 into a form of acid that can dissolve inside your plasma. Okay, so that's what we did last week. Let's move on to mechanisms of hemostasis. There are 3 ways our body uses to prevent ourselves from bleeding out profusely. They're very critical. called antibodies. Nice, and then you shout so loud. The mechanisms of hemostasis. What are the three mechanisms of hemostasis? We start number one. Teacher? Sorry? Oh, okay. Fibrin is one of the proteins involved in one of the steps. Okay? The step that you are referring to would be blood clotting. Okay, in blood clotting, fibrin is one of the proteins involved. Okay, thank you. So fibrin is one of the proteins involved. There are two more methods that we use to stop our blood from flowing out of ourselves. Henry? Dave, I asked you already, right? Okay, never mind, thank you. Okay, so vascular spasm. Vascular spasm. Do you briefly describe what vascular spasm is? Okay, contraction of the smooth muscles surrounding the blood vessels. Okay, if this is your blood vessel, and if you are damaged over here and blood is trying to leak out, the best way is that we constrict the blood vessel right before using the muscles that are surrounding it. So vascular spasm. Number two, what other ways can we use to stop blood from going out of ourselves? What's vascular spasm? Second, platelet plug formation. Okay, we make use of one of the components of blood, platelet plug. As the name suggests, all it does is to really plug up the patch. This only works if the damage is small. If it's too big a damage, platelets won't serve that useful a purpose. Instead, you may need to go for surgery to stitch up the damage if it's too big. Last is blood clotting. Last week, I got you as a class to complete this table for blood clotting. Yeah? I hope that by now that table is filled. This table you cannot find in your textbook. Perhaps I'd like you to screenshot whatever you've done. You can put it into your notes beside a paragraph where they talk about blood clotting. Or if you're the kind of student that revises both the CA and the notes, then you don't need to do that. But if you're the kind that only revises the notes, then you may want to transfer some stuff over. Okay, I'll not run through the various components needed in blood clotting, but I'd like to remind you that you are expected to know 1, 2, 3, 4, 5 of the components that lead to blood clotting. Okay? In a correct sequence. That's expected of you. Today we're going to move on from last week, hemostasis. Hemostasis is about one of the methods our body uses to protect ourselves. Okay? This is for protection at the end of the day. We want to protect you from losing too much blood. We want to protect you from bacteria, viruses that are trying to enter our blood system. Are there other ways that our body protects us from bad things from the surrounding environment? There is. And today we're going to focus on that. Today we're going to look at two other components. Namely, number one, the white blood cells. Number two, we're going to look at the proteins that can be found in our plasma that aids in protection. That's our focus for today. First and foremost, we're going to do an introduction to the various kinds of white blood cells that you can find in your body. If you scroll down, you can find like a family tree of white blood cells. We've left a lot of blanks for you. dedicate you now to go to your textbook, notes on cardiovascular system. And if you scroll all the way down, there will be a section of white blood cells. I'd like you now to do a quick read of that page, extract out and categorize all the white blood cells into that family tree. Okay, I'd like you to read page 29 and 30. 29 and 30. You don't have to do this yourself. You can work with your friend. Highlight all the different kinds of white blood cells you can find and categorize them into that chart. That will serve as a very good summary for you. You don't have to read that paragraph again.
example for monocytes. Then another type of monocyte. There isn't, but monocyte can mature into another kind of cell, such as MA. Do you find that cell? Let's summarize what you have. Alright, first and foremost, what are two broad categories of white blood cells? Hai. Okay. Granulocytes. A granulocyte. Granulocyte, they're called granulocytes because the white blood cells come with a lot of small little vesicle-like structures. They look like granules. Those granules actually contain a lot of enzymes inside. Very useful for digesting bad stuff. We call the other kind of white blood cells agranulocytes because you don't find that many granules inside. So you don't see those little dots. If you ever were to look under the microscope and you can see white blood cells, granulocytes come with a lot of dots. Agranulocytes don't really come with these dots. Okay? The word A means that it doesn't come with. Okay, let's look at granulocytes. There are three kinds of granulocytes. Three, yeah? Okay, neutrophil, Eaton, sorry, basophil. Okay, basophil. Okay, last one. Alpha, eosinophil. Okay, three kinds of granulocytes. I do not need you to know the function of each one of these granulocytes. Okay, just a general idea of granulocytes will do. Know the names, but I do not need you to go and look in depth into each one's function. Okay, they perform slightly different functions, but just a broad one will do for granulocytes. Okay, the idea that they contain a lot of chemical-filled vesicles. Okay, then you see the bullet points 1 and 2. That they are able to release enzymes and able to phagocytose. Okay, the word phagocytose, we're going to look in depth in a while. Next, let's look at agranulocytes. We have two broad categories of agranulocytes. The first one, let's look at we have monocytes. And you read, monocytes can transform into another kind of cell that will be really good at eating up other cells. And we call that cell trafficker. We do not call that cell trafficker. Macrophage. You see the word macro? Macro means kind of big. Phage means to eat stuff. Next kind of agranulocyte, Wu-Zhi-Xuan. Sorry? Yeah, you got it. Just correct. Yeah, lymphocytes. Yeah. Lymphocytes. They're called lymphocytes because they're named after the organ that produces your white blood cells. Do you know your lymph nodes? Okay. Have you ever fallen ill, then the side of your neck starts to swell up? In fact, right now, if you feel the side of the neck, you try to find a lump. There should be lumps on both sides of the neck. Have you? It feels like a oval-shaped lump. Or am I the only one that I need to go to doctor check it? Huh? Do you feel like this nice little like oyster inside, right? Yeah? Those are your lymph nodes. Next time you are sick, like you have a fever or something, when you touch the same organ there, you'll find that you'll swell up around 2 times the size. Because at the lymph nodes, they are producing a lot more of these white blood cells to go and defend your body. Okay? Go and feel next time. Every time I have a fever and I touch my lymph nodes, right, it's like swollen. Okay? There are 3 kinds of lymphocytes that you can find. 3, yeah? Okay? First one, natural killer cells. The name itself really suggests it does quite a decent job at killing foreign invaders. Shall we give him the last 2? Okay, thank you. T cells and B cells. Okay, 3 kinds of lymphocytes. Once again, I do not need you to know in depth all of these cells. Okay, just being able to state that there are 3 kinds of granulocytes, that monocytes can turn into this, and there are 3 kinds of lymphocytes will do. When you go to SH, then you learn all these cells in depth. If there are any cells out of all of them that you want to pay more attention to, would be B cells. Okay? B cells, as we will find later on, are responsible for producing very special proteins to help defend our body against bad stuff. I also like to pay attention to macrophage. Because the macrophage, as you find in your textbook, the paragraph is quite long. In this section over here, macrophage, quite thick paragraph. Macrophage can carry out a function that I think you have learned before under the chapter of membrane. Okay, and the word is phagocytosis. Phago means to eat. Cytosis means cell. That means to eat cells. Okay? And macrophages are really good at that. And let's have a look at the idea of phagocytosis now. Macrophage. Anyone remember the word phagocytosis? Was this totally foreign to you? Okay, if you flip back to your chapter on transport across membranes, this word is there. You can refer to this diagram over here. Have you ever wondered what's the purpose of lysosomes? Well, it serves a function to complement the process of phagocytosis. Supposing this is your cell, and supposing there is something really bad. Okay, I think I brought a magnet of bacteria. Okay. Oh. Okay, here we have a bacterium. This bacterium is not very good, fella. And this is your cell. Macrophages are able to do as it says. It can eat other stuff for the purpose of digesting them. So this is what a macrophage will do. Number 1, you've learned the process of endocytosis. That's the first thing it will do. It will start to endocytose the bacterium. This bacterium will then later on be fully engulfed inside the vesicle. But then you may be wondering, keep inside what? Like parasitic babies within it. No, it doesn't do that. And that's where the lysosomes come in. You learned before that our cells are able to produce a lot of vesicles called lysosomes, inside containing lots of enzymes. Hydrolytic. Hydro means water, lytic means to break. That means enzymes that use water to break stuff. Hydrolytic enzymes. These enzymes come in really useful because when we have these 2 vesicles fusing with each other, then all these powerful enzymes to go in and digest the bacteria, destroying it in the process. Yes? Oh, this entire cell that's smiling, right? Yes. So this entire cell is a macrophage. A macrophage is a kind of cell that specializes in eating other stuff. Okay? Yes? The macrophage contains lysosomes. Lots of lysosomes. And you've learned this organelle before in under the chapter of cells. Yeah? We even learned how the proteins inside are made. Okay, lysosomes. So we can relate back to a chapter you've learned before. A number of our immune cells, the white blood cells, are able to carry out phagocytosis. They're able to eat up other cells. Yes? Okay, and? Oh, why does it eat the entire thing, is it? Partial. Oh, side P also. Oh, okay. I think in one of your previous chapters, you may have encountered a word phagocytosis. Anything else? Okay. Phagocytosis is a form of engulfing where you engulf liquids only. Okay? Phagocytosis is a form of engulfing where we only engulf liquids. No cells involved. Okay? This was from one of your previous chapters. If you look into your textbook and give it a quick read, you'll find that granulocytes are also capable of carrying out phagocytosis. So how does our blood protect us? Because we have white blood cells that can carry out phagocytosis. We have white blood cells that can quite literally eat up the bad stuff. For those of you that have gotten an injury before, and then you find at the region where you got injured, right, it starts to swell up. Experienced that before? Yeah? So the region where you got injured, it may start to swell up. So supposing maybe this is your arm. Okay, then it's starting to swell up in this region. Yes? The bacteria, is it? Yeah. So the lysosome would fuse with the vesicle that contain the bacteria, and then all the enzymes inside can digest it. Yeah, the big smiley thing was a macrophage. So if you look inside, if you look at this image here, this large purple cell, there's the macrophage. Yeah, that is digesting. If we can watch a video of a cell that is phagocytosing another one. Are you overseas? Okay, here you see lots of bacteria, and then you see macrophages coming in, literally just gobbling up the bacteria. As it gobbles it up, it also digests it using all the lysosome enzymes. See, the bacteria is gone. All the lysosomes have fused to the vesicles and release their enzymes. Can you see that it is slowly wrapping itself around? Okay, the bacteria has just been engulfed. See all these small little vesicles going towards the bacteria? These vesicles are the lysosomes containing all the enzymes. And once it fuses with this big pouch over here, it releases all the enzymes in to digest. Neutrophil. Neutrophil is a kind of granulocyte capable of carrying out phagocytosis. So once again, I do not need you to know each kind of granulocyte what it can do. Just broadly, they are able to phagocytose or do. Okay, so that's what phagocytosis looks like under a microscope for real. Yes.
So all granulocytes can like undergo phagocytosis.
Yeah. Yeah. Okay. Okay, so that's the first method of protecting ourselves. We just eat up our foreign invaders. Yes. Yeah.
Can they eat each other?
Oh, you mean like could these cells eat each other?
Yeah, if one of them is sick.
Oh, can. So granulocytes, macrophages, they're able to eat up cells that are also not doing so well. Yeah. But if you want to eat up cell to cell, actually it's quite big. So normally what cells will do is they'll first cause them to die. Then after they die already and they start to disintegrate, then they eat up the parts. Okay? Now the next class of white blood cells that I'd like you to pay a little bit more attention to are your B cells. Your B cells are able to produce a special protein that can serve a very important function in our immune system. Which now brings us to the next part of the chapter, antibodies. Let's go to the next part of the CA on antibodies. Our B cells are able to produce a protein. So again, I'm going to emphasize that this is a protein. Okay? Antibody. Body, if it is singular. Antibodies, if it is plural. So singular, plural. And again, I want to make an explicit link. Antibodies are proteins. That means to say they have primary, secondary, tertiary structure. And I'm again going to tell you in your face that antibodies go up to a quaternary structure. Just like hemoglobin. Okay? So a clue for you. If we ever show you this image of an antibody, we ask you to describe the levels of structure and save one level of thinking because it has a quaternary structure. Yes? And okay, anything that's a protein can denature. That's why it's not so safe if you have a high fever for too long. If you have a high fever for too long, a lot of proteins that may normally function at body temperature may start to unwind itself and lose their function also. Okay, antibodies are proteins. These proteins are not enzymes, but they are able to serve a protective function. Let's watch a quick video on antibodies as an introduction. video, I'd like you to take note of how many polypeptides a single antibody protein is made up of. Then at least you are a little bit more aware, when we say it's quaternary structure, how many polypeptides there are.
If this is the first time you've heard about antibodies, you may not know how important they are. Antibodies are one of the warriors inside our body helping us fight off nasty invading particles. They are a type of protein and a key component of our immune system. If a person doesn't produce or overproduces antibodies in the body, the person will become very ill. So where do antibodies come from? Antibodies circulate in the bloodstream and can occur anywhere throughout the body. Their function is to recognize bad agents called antigens and will trigger further chemical reactions in the body to remove the antigens. Antigens are foreign substances that are capable of triggering an immune response. They can be viruses, fungi, bacteria, and some non-living substances such as toxins, chemicals, and foreign particles. They can even be allergens like dust, pollen, or certain foods that cause allergic reactions. Antibodies are produced in the immune system in response to the presence of an antigen. So what do antibodies look like? Antibodies are also known as immunoglobulins. They are generally a Y-shaped protein consisting of 4 polypeptides, 2 identical light chains, and 2 identical heavy chains. The terms light and heavy simply mean larger and smaller. The 4 polypeptide chains are connected by disulfide bridges. Here, at the tip of each arm are the antigen binding sites. These are really important parts of attacking invading antigens. Similar to the lock and key mechanism in enzymes, the shape of the antigen binding site is specific to the antigen it's targeted. If part of the antigen fits into the antigen binding site, the antibody recognizes the foreign antigen and binds it in. This stops the antigen from causing more damage elsewhere. 2 identical binding sites means any given antibody can bind simultaneously to 2 identical antigens. Millions of antibodies with slightly different tip structures exist, with each variant binding to a different antigen. This enormous diversity of antibodies allows the immune system to recognize an equally diverse range of antigens. By binding to an antigen, an antibody can stop the antigen from entering or damaging cells. This is known as neutralizing. The antibody can also fight the antigen or attack by other parts of the immune system. In mammals, there are 5 immunoglobulin isotopes. They perform direct diols and help directly appropriate immune response for each different type of foreign invader. Antibodies are cloned in laboratories. To discover how and why, watch this video.
Okay, how many polypeptides? 4. Okay, our antibodies are quaternary proteins. They're made out of 4 polypeptides. They are held together by a bond that you've learned before that holds tertiary and quaternary structures together. This I need you to take note. Disulfide linkage. Okay? This is not out of your syllabus. We've learned before that quaternary structures can be held together using this bond. Okay, disulfide linkage. Okay, and as the video suggests, it's made up of 4 polypeptides. This is a quaternary protein. Shall we practice as we normally do if we see a protein? Look at this protein now. Can you describe the levels of structure? We do a quick revision now. Okay? Can you describe the structure of this antibody? How will you describe each level? From primary to secondary to tertiary. Okay, and then there are always very typical vocabulary you can use to describe every single structure. You look through any one of your worksheets, we will do it kind of the same way. together in bullet points. Okay? Levels of structure in an antibody. First level of structure, we call this structure the Ella? Primary structure. Will you elaborate a little bit? For primary structure, how will you describe the primary structure? What does it refer to when we say a primary structure? Thank you, Ethan. Sorry, you say sequence, right? Okay. The sequence, the number of amino acids, the type of amino acids present. Okay. That's what primary structure is referring to. I'm going to write in red, in black, all the interactions involved. What kind of bond is involved in primary structure? Yeah, peptide bonds. Okay, that's what's holding the amino acids together. Maybe read one more thing. Okay, your peptide bond. That's what's holding your amino acids together. Then we progress to the secondary structure. If you were to describe the secondary structure are localized holdings. Localized means that the folds, you find them in selective regions. And there are two kinds of secondary structure. Sorry? What are the two kinds of secondary structure? The alpha? Alpha what? Okay, alpha helix. Anything else? Okay, thank you. Beta pleated sheets. That didn't sound like soya. Pleated sheets. Okay, and what kind of bonds are holding the secondary structure together? Suji? Okay, thank you. Hydrogen bonds. By the way, this can apply to any protein that we've learned so far. Most recently, we learned about hemoglobin, right? We apply this also, and we also learned about carbonic anhydrase and enzyme. Also can apply these concepts. All the levels of structure. So this is a very useful thing you want to revise, because any protein you find in your body likely can be described this way. Tertiary structure. You can describe it as having a 3D globular shape with large scale folding. What kind of bonds are involved in tertiary structure? A lot. Okay, from your disulfide linkage to your hydrogen bond to your hydrophobic interaction to your ionic bonds, okay, all the usual bonds. And let's say we were to give you an antibody, then it will go up to a quaternary structure. Okay, so you take note what kind of protein is provided to you. An antibody is provided and you got quaternary. Means that there are more than one polypeptide. And the kinds of bonds that are involved will be the same kind of bond that you will find at the tertiary level. But just in this particular example, the main bond involved is disulfide linkage. Okay? If you want to prep yourself, for any protein And whatever protein we teach you, you just quickly Google and find out it goes up to which level of structure. I think we'll not do blood types today. To end up today, we're going to talk a little bit about how antibodies work. If you're not done jotting down whatever you want to jot down, so I get you to just snap a quick photo. Okay, we're going to erase the left side of the whiteboard. Okay, to end up today, let's look a little bit about how antibodies work. They function in several ways. To understand how they work, we need to understand how the structure plays into its function. First and foremost, you find that there are two big chains. These two big chains are called heavy chains. The two smaller chains are called light chains. Combined together, they create a special region that can bind foreign substances. Okay? This region is called an antigen binding site. The ABS, antigen binding site. Therefore, there's one new vocabulary we're going to learn today. The word is antigen. Antigen refers to anything antibody can bind to. It is usually foreign. When I say foreign, I don't mean of another country. I mean not of your body. Okay? Antibodies can bind antigens. And antigens are usually foreign in nature. I'll give you an example of an antigen. Here I have bacteria that has just entered your system. These bacteria, just like any cell, also come with cell membrane, and they'll come with proteins, membrane proteins that are embedded on the surface, represented by this triangle black stuff. Okay, I'll pause a while. Tell you all to taking notes. Okay, we're more or less done. Okay, so back to where I was. I was saying that, for example, if a bacterium enters your system, you find that, an example, bacteria comes with cell membrane, and the cell membrane comes with a lot of proteins on the surface. These proteins could be antigens also. Antigens are anything antibodies can bind to. Okay, I say again, antigens, anything that antibodies can bind to. And they are usually foreign in nature, not from your body. So for example, here I'm going to plop in several antibodies produced by the B cells. If you have something foreign that enters your body, these antibodies will be able to bind to the antigens of the bacteria. Notice that they function kind of like how enzymes do, complementary. Okay? The antigen binding site is often complementary to the antigen it needs to bind to, just like an enzyme. But bind for what? Like a bit useless, huh? Just by binding, what purpose does this serve? Okay, there are several reasons why antibodies will want to bind to antigens. Number one, let's say if the bacteria wants to infect another cell. Okay, so I have a poor cell over here that is about to be infected by bacteria coming in. Yeah? But right before its cell membrane is able to attach to the cell membrane of this cell to fuse and enter, here comes the antibodies, attaching itself to the outside. Now, even if you wanted to fuse and enter the cell, you cannot enter anymore. Yeah? So that's one function of antibodies. They help to, as the words, one word you can use, neutralize. They help to neutralize any foreign substance. Yes. Okay. Charmaine asks, is this bacteria itself an antigen, for example, right? Or are the proteins on its surface are antigens? Okay. So, in this example, the proteins on the surface are the antigens because that's what the antibodies bind to. Okay. Let's say this bacteria is able to produce certain chemicals that are toxins. The antibodies can also help to neutralize the toxin by binding to the toxin. If it binds to the toxin, then the toxin cannot enter the cell. Okay, so there's one way antibodies can help to protect you. It just wraps itself around whatever thing is bad. Yes.
The toxin and antigen now.
Oh, yes. So the toxin in this case is the antigen. So antigens are, that's why I put anything that an antibody can bind to. It's a very broad category of things. Okay, so we can neutralize a lot of things. Neutralize toxins, neutralize anything bad that wants to enter. Okay, and before we end off today, one last method. It's called opsonization. Opsonization. Antibodies can also, yes.
So the entire process of binding to the antigen is called neutralization.
If antibodies bind and wrap around something bad to prevent it from entering another cell, that's called neutralizing. Another method of protecting you is called opsonization. When antibodies bind to, let's say, foreign substances, sometimes it helps to, and let's say we want to eat up these foreign substances. Let's say now we have a macrophage. These antibodies actually have receptors. Sorry, there are receptors on the surface of macrophages. And these receptors can detect the antibodies. Once detected, it will just engulf the entire thing. Okay? So, antibodies can serve as also signals. Antibodies can also serve as signals for macrophages. We call this process opsonization. When antibodies serve as signals for macrophages to detect, that's what opsonization is. So kind of two ways now. One is to neutralize, you just engulf the entire thing with antibodies to protect the cell from it. Number two is to serve as signals so that cells that can phagocytose will be able to detect. There are many other ways that antibodies can defend you, but I'll just introduce two ways. Yes. Opsonization what? Will produce enzymes. Oh, no, its main purpose is to help macrophages detect the bacteria. So when the antibody binds to bacteria, then the macrophages have receptors that can detect antibodies. So they will help to engulf from there. So that's what opsonization is. Neutralization, is it? After it binds, is it? So after it neutralizes, it could then be detected by macrophages to engulf. Is it step 2? I would say it's kind of hand in hand. Only if a macrophage go and detect it, then I would say there is opsonization taking place. Okay? Okay, that's where we'll cut off today. You have math now? How come your math teacher not here? Okay, please move on to your various rooms. Okay, that's all for today. Thank you.
啊，我食邊人就算買。